Thank you so much for this opportunity. It's really a pleasure to be presenting in this ISCB Academy for the first time. As Alice mentioned, my talk is about deconstructing and reconstructing microbiomes and understanding their role in antimicrobial resistance transmission. So the problem statement is really the growing challenge of antimicrobial resistance. As probably many of you know, there's rising rates of multi-drug resistant infections in different parts of the world, and particular in Asia, where in some countries, resistance to all common antibiotics is going up, and for some species, the resistance rates above 50% in terms of the strains that are observed clinically. And this, if it is left unchecked, could lead to potential collapse of healthcare worldwide because microbes don't see borders. And it's estimated that by 2050, we could see as many as 10 million deaths per year. This is more than all cancers put together. And this could have a significant impact on healthcare systems as well as economies worldwide. Now, my lab at the Genome Institute of Singapore in A-Star is Laboratory of Metagenomic Technologies and Microbial Systems. And more specifically, we are interested in developing technologies to understand human environment interactions, particularly in the context of antimicrobial resistance transmission between these two sources. And for this, we think we need new technologies for both assembling microbial communities and understanding their constituents, but also in terms of modeling how the community is assembled and how the constituents of the community interact with each other. We are interested in studying the skin microbiome as an important interface between the environment and human beings. We worked on studying the gut microbiome and how antibiotics perturb the gut microbiome and how that can lead to colonization by multi-drug resistant bacteria. And finally, we've been looking at various environmental reservoirs of antimicrobial resistance, particularly in areas of importance for human habitation, particularly areas like hospitals, food courts, and we've been involved as part of the MetaSub consortium to study environmental microbiomes in general. So currently, when we think about microbial surveillance, the challenge really is that the clinical workflows that we have worked on over the years are very pathogen specific, and they're also very resource intensive, requiring multiple steps from culturing to mass spectrometry to PCR to validate the presence of an organism. Of course, those of you who are in genomics know that metagenomics is the potential to provide a unified workflow that could allow us to assay and survey diverse microbes with a single workflow. And there are various metagenomics workflows that have been used primarily to study microbial communities, and not as surveillance workflows, including 16S ribosomal RNA sequencing, and of course, shotgun metagenomics. But both of these workflows perhaps is too low a resolution if you want to think about microbial surveillance, which is traditionally being done through isolation and whole genome sequencing, with the whole genome of an organism serving as the critical information for understanding transmission patterns across environments. So for many years now, we've been excited about the potential for long-read sequencing in being able to deconstruct a metagenome into its constituent genomes. And nanopore sequencing is particularly exciting because it's a portable, easy-to-use technology, which has traditionally been seen as a capability that even infectious disease labs in small hospitals can deploy. And what we have been looking at is really the ability to use nanopore sequencing in clinical workflows. So one piece of work that we did a few years ago is to show that we can get nanopore libraries and sequencing with high throughput as well as high N50, so in the several kilobase pairs range, for clinical samples such as stool samples that we would like to do surveillance on. But the next challenge, of course, becomes to use that data. And at that point of time, when sequence error rates were still high, we conceived of a metagenomic assembler that leveraged the strengths of short-read sequencing, the accuracy of short-read sequencing, with the long contiguous information provided by long reads, and to think of metagenomic assembly as a decomposition problem of converting the graph of assembly into clusters for individual genomes that can then be assembled using single isolate genome assembly tools and techniques. Of course, we also wanted to leverage the fact that there's a lot of reference genomes that have been published by now, and these can serve as at least an initial starting point to guide the assembly algorithm. So this new assembly tool that we call Opera-MS is based on some very fundamentals of genome assembly, and I'd like to refresh your memory about that. So genome assembly fundamentally is the idea that we can take sequencing reads that come out of a common DNA high-throughput sequencer and recognize the fact that the end of a read is similar to the beginning of a read, as an overlap, which then serves as the way to bring these two reads together, just like pieces of a puzzle. And we can draw a graph that we call an assembly graph, where reads are nodes and edges capture overlap information. And then potentially a path through this graph gives us the genome of interest. Of course, this is a simplification of the problem, and this has been studied mathematically for many years now. And it falls into a favorite class of problems for computer scientists, which is NP-complete problems, and often is seen as a cop-out for computer scientists to say, well, this is a hard problem according to many other famous people, so we really can't solve it. And in fact, various sub-problems assembly, including the well-known scaffolding problem, the problem of taking a bunch of contigs or contiguous sequences assembled from short reads and aligning them and using mate pair information to align them and construct longer sequences called scaffolds, has also been seen as an NP-complete problem. What we showed nearly a decade ago is that despite it being an NP-complete problem, we can actually solve this problem optimally. And this arises because assembly graphs are not arbitrary graphs. They are actually graphs with specific structure imposed by the sequencing process, and we can exploit this structure to optimally solve the assembly. And so this we thought was a very useful building block because it's an isolate genome assembler, and we want to now decompose metagenomic assembly into its constituent parts and then apply an existing tool to address that problem well. So the problem for us eventually became, how do we optimally cluster contigs for a metagenome? If you imagine contigs for a metagenome as being nodes in this graph here, and the color indicates the coverage, how many reads cover a certain contig, then contigs that have similar coverage potentially come from the same genome. Here the colors perhaps also indicate, the shapes perhaps also indicate the origin, the species origin of these contigs. And we can use a simple clustering algorithm purely based on the assembly contiguity graph that we have at the end of any assembly to cluster these contigs. But then we still want to cut this graph based on the coverage of contigs and find cuts where the contigs that have very similar coverage are brought together. And so we formulate this problem as one is of finding a cut which optimizes a well-known information criteria that you are probably aware of, Bayesian information criteria, which balances the model complexity with the number of parameters of the model, K, which is the number of clusters that you determine. It turns out that this problem can also be solved optimally using a recursion approach, and it's quite a simple mathematical derivation to convert this information criteria and optimization question into a recursion problem. And so this formed the basis of an algorithm that we called Opera-MS. And Opera-MS, with its attempt to do optimal assembly and leveraging long reads, was clearly an improvement over short read only assemblers, so Illumina assemblers. And here I'm showing a few different assembly algorithms on the x-axis, and on the y-axis is the percentage improvement shown by Opera-MS. And not surprisingly, it was almost an order of magnitude improvement. You have much longer reads, and so you expect better assembly. But compared to long read assemblers, which were available then and not specifically designed for metagenomics, we found that we still got a substantial boost, nearly 200% improvement in assembly contiguity. And this really was a function of how much coverage you had for a particular genome that was being analyzed. But finally, the most important aspect of Opera MS was that it can do strain level assembly. Perhaps it was the first metagenomic assembler that showed that you could disambiguate perhaps two different E. coli strains which are there in the same metagenome. So what can you do with such an assembly algorithm? We first wanted to show that even in complex stool samples, we can learn something interesting. And in this case, we looked at all the circular contacts that we acquired, and they spanned a range of contact lengths all the way from 5 kb to 200 kb. When we looked at the similarity of these contacts to known sequences, we found that there's a small number of contacts which are very low identity and fraction covered compared to known sequences. And it turned out that one of these sequences, which is circular sequence, is well annotated with genes which are known to be important for phages. And we speculate that this is a jumbo phage genome that was the first time assembled directly from the human gut microbiome. But more specifically, beyond understanding biology, can we use it for applications such as surveillance in hospitals? And in hospitals are an important area of concern for environmental transmission, because traditionally infection control in hospitals has focused on patient-to-patient transmission. But it's well known that the environment also serves as an important intermediary to transmit pathogens from one patient to another. So it's an unknown but significant contribution. And using a metagenomic approach, we can potentially get a more comprehensive view of all the microbial genomes that are present in hospital environments. And so to do this and to show a proof of concept, we envisaged what we called a smart hospital project. And the idea here was that if this works out, we could do this as a more routine operation in hospitals to make them smarter, to do infection control in a data-driven manner rather than in a blind or in a almost carpet bombing manner. So the project design involved selection of various hospital wards, all the way from isolation rooms where there's only a single patient warded to standard rooms where there are many patients, as well as MDRO where patients who are colonized by MDROs or multi-drug resistant organisms are warded. And we selected sites which are typically high-touch sites or are sites of concern for infection control, such as sink traps and sink basins. And we used a combination of Illumina as well as nanopore metagenomics to intensively interrogate the metagenomes in about 355 sites, 179 sites and 355 microbiomes, where we had 2 time points from each site initially, and then a third time point that came more than a year later. And we generated about 30 million reads on average. And then we use this information to construct metagenomic assemblies. And what was striking was, because these communities are not as complex as environmental communities, and we have a quasi-metagenomics angle where we culture some of these species and do a plate sweep, we were able to get very high quality genomes. So for most pathogens of interest, we were able to get N50s greater than 1 megabase pair, which essentially means the genome is in a few discrete contexts. So once you have a situation where the genomes are in a few contexts, this allows you to then do the traditional kind of analysis where we compare genomes, whole genomes, and construct phylogenetic trees. So we did this for Staphylococcus aureus, for all the Staphylococcus aureus genomes that we acquired from the hospital environment, and they were clustered at 99.9% identity to represent strains. And we found that there's quite a bit of diversity of Staph aureus genomes. And in fact, there are several strains which seem to be resistant to all 5 different antibiotics that we tested phenotypically. And when we look at these multi-drug resistant bacteria and the specific strains here, S3, you can notice that S3 seems to be also very dispersed throughout the hospital environment. So across different wards and across different sites in the ward, we find that S3 is again and again observed and present at the genomic level. And we see this across multiple time points as well. Now, because we used a metagenomic approach, we have genomes for other potential pathogens as well and other species as well. And so we can check for this pattern and again. And so we can see that across the pathogens that we had sufficient number of genomes for, we see a recurrent pattern. The more multi-drug resistant a strain is, as for example, for Staph epidermidis, there's S16 and S1, which are multi-drug resistant, and both of these strains are in fact the more widely distributed and widely persistent strains as well. So this is a recurrent pattern in the hospital environment. Now, unfortunately, in this study, we did not have ethics approval to collect patient samples, but we were able to leverage the fact that there's routine surveillance studies frequently in hospitals. And in this case, we had an MRSA, methicillin-resistant Staph aureus, surveillance study that was done in the hospital more than 8 years ago. And taking the genomes from these strains, we compared them to the hospital environment genomes. And we were very surprised to find very similar strains, some strains with as few as 5 SNPs between them, and many strains with less than 50 SNPs between them, suggesting that there's remarkable genomic stability and potential reservoirs in the hospital environment that could explain this overlap. But in addition, we found that the overlap was enriched for multi-drug resistant strains as seen here, S1. And again, because we had genomes for many different species, we could check this pattern again and again and again, and we could find a statistically significant enrichment in the overlap of hospital environmental strains and patient strains for multi-drug resistant bacteria. So this was a concerning factor and pointed to the fact that we might actually be enriching environmental reservoirs of multi-drug resistance in hospital environments, potentially due to the cleaning measures, but also potentially to seeding effects which might seed colonies and reservoirs in the hospital environment. Another area of concern in terms of reservoirs for antimicrobial resistance is the gut microbiome. And as those of you who study this community know that it's an incredibly dense environment, also diverse environment in terms of bacteria. And so the various mechanisms that we know for transfer of resistance genes, including conjugation and transaction, can operate more frequently in the gut environment and provide a mechanism for transfer of resistance. So what we wanted to understand is how can we prevent transfer of resistance and how can we stop colonization and persistence of multi-drug resistant organisms in the gut microbiome. And as you know, one of the important risk factors for colonization by resistant bacteria is in fact the use of antibiotics in the first place. So what survives antibiotic selection might actually already be drug resistant and then persist and then further lead to transmission as more antibiotics are used. And so to look at this phenomena, while people had noticed that antibiotics can perturb the gut microbiome and can leave it perturbed for extended periods of time, what we didn't understand is why was the perturbation of the gut microbiome different in different individuals. So it has been observed that in some individuals, the microbial diversity goes down during antibiotic usage and comes back up post antibiotic usage. But in other individuals, diversity remains low and stays low for long periods. And so leveraging the fact that there's been many published cohorts for this subject, including our own study, which was a natural history study in the sense that we were recruiting subjects who were naturally were taking antibiotics for treating infections rather than healthy individuals. We used all of this data in a cross-validation framework to identify bacteria in the gut microbiome that might associate with recoverers or non-recoverers. And because we had multiple cohorts, we could find robust signals across these various geographical regions, which again and again associated with recovery. We found this short list of 20 species had many Bacteroides species, a few Bifidobacterium species, and a few Patabacteroides species, showing that there's consistency in the bacteria that are associated with recovery. I won't show you all the analysis that we did in here, but we also showed that potentially there is a process of cross-feeding and almost a food web network in the gut microbiome that some of these species could seed and therefore promote recovery of the gut microbiome. But here I'm showing you an example where we used a mouse model to validate some of these observations. So in this case, we took some of the recovery-associated bacteria that we call RABs, including Bacteroides theriadomicron and Bifidobacterium adolescentis, and we gavaged these two species that were identified as RABs, as well as a combination of them to test for synergy, as well as a vehicle control. This is work done by Dr. Yihan Tan, and monitor the recovery of the gut microbiome over many days. And one of the striking observations that came out of this study was that, in fact, Bacteroides theriadomicron, or BT, leads to 100-fold higher recovery of bacterial biomass just a few days after antibiotics. And BT alone does it, but BA, which is Bifidobacterium adolescentis, does not. But in combination, BT and BA promote alpha diversity in the gut microbiome. So they lead to higher diversity. So there's a synergistic interaction between these two bacteria. While Bifidobacterium adolescentis, which is a common probiotic, does not in itself promote biomass recovery, it promotes diversity in conjunction with Bacteroides theriadomicron. So after, let's in this situation where the microbiome recovers, of course, we would hope that colonization resistance sets in and prevents the colonization of multi-drug resistant bacteria. But often this may not happen, and so when recovery fails, we would like to understand the mechanisms by which carbapenemase producing Enterobacterales, which is a important class of multi-drug resistant bacteria, can persist in the gut microbiome. And so for doing this, we were leveraging the fact that in studies in Singapore, we found that in fact 90% of individuals actually spontaneously decolonize within a year. So there are certain gut microbial factors or other factors that perhaps promote decolonization. And by doing a longitudinal study on a subset of individuals who have colonization, we can understand perhaps the factors that lead to decolonization. So we recruited 29 index subjects who were colonized by carbapenemase producing Enterobacterales, as well as their family members as controls. And we used gut microbiome sequencing, so metagenomic sequencing, as well as isolate sequencing of the CPE species, so the E. coli and the Klebsiella, which are multi-drug resistant here, as part of the study. What we wanted to study was both the microbial ecology, but also at the strain level, the dynamics of E. coli and Klebsiella, as well as genome level evolution of the various strains and acquisition of resistance, perhaps over time. So at the ecological level, what we found was that if you took the gut microbiome of these various individuals and projected them onto these first two principal components, we see a region, we see 4 different clusters, but 2 of these clusters are more dominated by family members, so the healthy subset of our cohorts, while the colonized individuals shown in red here tend to be enriched in clusters 3 and 4. In fact, if you just do a statistical test, you can see that along the first principal component, in fact, there's an enrichment and a shift to the left for CPE positive individuals. So when they're individuals at the time point in which they're colonized by carbapenemase producing Enterobacterales, and this goes away as they're decolonized, and it fully recovers by the time they are by the time they resemble more their family members. So at the decolonized time point after several months. So when we compare these communities, of course, in addition to the shift, we can identify several species which are differentially abundant. And but the short list of species that we identified, in fact, seem to be enriched for species that have immunomodulatory functions. So after decolonization, the recovery of specific species seems to lead to immunomodulatory function from different pathways. And many of these species were also identified as recovery-associated bacteria in our original study, including Faecalibacterium prausnitzii, which has an important role as a butyrate producer in the gut. But also if you look at the metagenome and the genes and pathways that are enriched during colonization, we see an enrichment for inflammation-associated pathways through various mechanisms, from sulfate reduction to aerobic respiration and oxidative phosphorylation, which serve as signatures for inflammation in the gut and the response of the microbiome to that. So overall, it seems like we have a perturbed gut microbiome, even though the colonization by carbapenems producing Enterobacterales is asymptomatic and does not lead to overt disease. But this colonization is also not very stable. So what we find that if you look at the abundance of Enterobacterales in the gut microbiome, the relative abundance is very high in some individuals, as high as 80 or 90% can be dominated by E. coli, Klebsiella, and other species. But also that this is not very stable. So, for example, in subject 0457T, where we are showing one stacked bar chart per time point, what you can see is that the community changes quite rapidly from one time point to another. In another individual, it switches from a Klebsiella dominant gut microbiome to an E. coli dominant gut microbiome and then back. And finally, in subject 15674T, we see co-colonization by both E. coli and Klebsiella pneumoniae. They're both enriched in the gut microbiome. But in general, the dynamics is quite unstable for Enterobacterales colonization. And we can see that comparing subjects and family members, that the standard deviation of abundance is greater for all these species. And in particular, when there is two species colonization, when there's E. coli and Klebsiella in the gut microbiome, we see an interesting example of plasmid sharing. So in this individual 1674T, we see that among the several plasmids that we assemble from the isolates, we find that there's a sharing of two key plasmids. One of these, PKPC2SG1, is the plasmid that confers antibiotic resistance to carbapenems in Singapore, and we find that both of these plasmids do not have a single SNP between E. coli and Klebsiella pneumoniae, so they're highly similar. And based on in vitro experiments and estimates of divergence over time in vitro, we estimate that these two strains likely shared the plasmid less than 6 months in time, so potentially during gut colonization. And we can also show evidence that the transfer of this plasmid between E. coli and Klebsiella pneumoniae is possible, and it happens at moderate conjugation frequency. So here's potentially an evidence for transfer of resistance, which is perhaps what we are most concerned about with gut colonization. So we really would like to figure out ways where we can prevent colonization of the gut microbiome. And in fact, natural unperturbed gut communities have an intrinsic resistance to Klebsiella, E. coli, and other Enterobacterales, as can be shown in this in vitro experiment where we do not, where it's done in a reactor community. We can see strong inhibition of Klebsiella pneumoniae when grown with stool communities. And this is work from Prof. Ganyu and Huen's lab at NUS, work done by Dr. Chiang Kai Cheung. But what we don't know is why this happens, which constituents of the gut microbiome promote colonization resistance. We know that there's complex interactions that happen in the gut microbiome, but we know very little about the specific interactions that happen. And while there have been several works on how to study this, we think there's scope for developing new and improved methods. This is what the lab has been focusing on for the last few years. So I'd like to tell you a little bit about these new methods that we have developed. So the first method that we call BEAM is to be able to infer really an ecological model of microbiome dynamics. And it's based on classical Lotka-Volterra models, in this case, a generalized Lotka-Volterra model, where the rate of change of abundance of species I, which is dxi dt, is a function of abundance of species I at time point t, multiplied by a growth rate parameter mu I, plus a whole set of interaction terms for species I and species j, so xi versus nxjt, with an interaction term beta ij. And so taking this model, we note that actually these models classically were defined to work with absolute abundance data or absolute density data. In fact, when you think of metagenomics, what we have is relative abundance data. So we can't directly fit these models. But instead, what we came up with, and this is work from Li Chen Hao, who was a graduate student in my lab and is currently at the Broad Institute, is that we can actually reparameterize this equation. So we can explicitly bring out M of t, which is the abundance or the biomass of the whole microbiome at time point t, and rewrite this in terms of relative abundance terms x tilde j. And then by subtracting a reference species, and this can be an arbitrary reference species, from the left-hand side as well as the right-hand side, we get a ratio of relative abundances. And so we no longer have M of t on the LHS, so it can be estimated directly from the data. And all the parameters that we need to estimate are on the right-hand side. And this includes the AIs, which are a reparameterization of the mu i's, the growth rate terms, as well as BIJs, which are based on the beta IJs, the interaction terms, and M of t, which is the biomass term. But if you can see this equation, it's clearly not a linear equation anymore. In terms of the parameters, there's the multiplication of these parameters. And so we used a well-known trick, which is to solve this via expectation maximization, where we solve for AI and Bij, assuming certain values for M of t in one iteration, and then and then using those estimates of AI and Bij to estimate M of t, and then iterating this process to convergence. So surprisingly, this method works. And we were also quite taken aback because we didn't expect it to work so well. Here's a situation where you are completely missing ideally an experimentally derived measurement and still able to estimate model parameters. And so here are some results, and these are based on simulations, but in our manuscript, we actually have some real data as well. But in terms of simulations, if you simulate ecological models with true biomass and then use that to estimate parameters. This is the relative error that you measure, which is in the first box plot here. And for growth rate parameters, which is the mu i's, the interaction parameters, the beta ij's, as well as interaction network structure, which is whether the beta ij is 0 or 1. And you can see that with the true biomass, you do reasonably well. You can control relative error, and you can get pretty high AUC for interaction network structure. And even without knowing the biomass, the no biomass column, which is what Beam does, we can still recapitulate what you can get with the true biomass, given sufficient data to model. Of course, the interaction network structure is slightly less accurate. Compare this to other approaches. For example, if you can renormalize your data with CSS, which is cumulative sum scaling, as well as TSS, which is total sum scaling, both of these approaches do not perform as well. And if you use a qPCR, which is 16S qPCR, as a measure of total abundance and as an experimental approach with one replicate or with 3 replicates, the error in these measurements, in fact, cascades through the learning of the model and leads to pretty high relative error for many of the parameters that we estimate, and can be as bad as random, as shown in this gray line here. So this to us showed that we can actually use some clever techniques to learn models even when experimental data may not be as perfect as you'd like it to be. But in fact, if you think more about this, we actually have a bigger challenge, which is that generation of cross-sectional data is a lot more convenient than generating longitudinal data. In fact, if you think about the gut microbiome, the dynamics of the gut microbiome might be at the hourly or in a 6-hour or 12-hour time frame, while sampling may not be as frequent in most studies. But even despite that, generating longitudinal data sets is relatively rare. If you look at the public repositories, this shows the enrichment of cross-sectional data sets over longitudinal data sets, and you can see it's still about 50-fold. So you're much more likely to find large cross-sectional data sets. So how could we learn dynamic models from cross-sectional data? Sounds crazy, but you have to make some assumptions. And if you make the assumption that a fair amount of your data is at or near equilibrium, then you have this nice phenomenon that dxi dt is approximately 0. If you assume it to be 0, you will obviously have some error, but if the error is random, you might still be able to learn reasonable models. Of course, you have to reparameterize in terms of relative abundances, and so you can do that similar to what we did earlier. And the same EM idea, the expectation maximization idea, works here as well in terms of learning the model. Now, the other challenge here, of course, is that you need to know which microbiome profiles that you have are at equilibrium, and those that are not at equilibrium or are significantly away from equilibrium have to be removed from model estimation. Also, it's possible that some of your data points are actually not even part of the same model. Let's say you have two enterotypes or data from two gut microbial enterotypes. You can't learn a single model for both of them using this approach. So you have to be able to remove data sets or profiles which do not fit the model that you're trying to fit. Okay, so beam static, which is the method we designed here, actually has a way to do that. And we compared beam static with the commonly used approaches with cross-sectional data, which is to use correlation approaches and various approaches to measure correlation to capture interactions. And what we found is that if the data that you have uh, are really from an ecological model, so they capture GLBM interactions, then correlation-based methods essentially are as good as random, while beam static is very close to what a true biomass-based measurements would lead you to. Not only that, we can actually use this approach to get some biological insights. For example, if you have data where some of the samples involve individuals who are taking antibiotics, then you can flag these samples which likely don't fit your original model with pretty high sensitivity, so with 88% sensitivity. And also, we can also really refine our understanding of enterotypes. So classically, enterotypes have been determined based on clustering, based on principal coordinates analysis plots, or through other clustering approaches. But what we find is that if you do that and you compare that to the ecological enterotypes that we learn by iterating the process of model building, then we find that the ecological models are actually quite distinct. So here they're shown in two different colors, and you can see that some of the blue data points, for example, are in the peak OA along with the yellow data points and might therefore be clustered with the yellow data points. But actually, ecologically, they are pointing towards a different equilibrium that they like to converge to. Okay, so finally, I'd like to leave you with a vignette on how we can take this further into a situation where the colonization has led to an overt infection. So
and sequencing we use to catch and track bloodstream infections. And classically, the bloodstream has been believed to be devoid of microbes.
And only in disease cases do you have signatures of bacteria. You can grow bacteria from blood. But increasingly, there have been studies based on various sequencing as well as other approaches that suggest that there might be metabolically active bacteria in blood. And of course, this can be confounding if you want to use sequencing to detect bloodstream infections. And so this concept of a blood microbiome has been taking root over the years, and we wanted to investigate whether this is really true. So for doing this, we leveraged the access to the precision medicine cohort in Singapore, where as a pilot study, they did 10,000 healthy volunteers where they obtained blood and did whole genome sequencing and also collected metadata. And our goal was to use the non-human reads in this sequencing data to be able to identify the microbes that might be present in blood. But of course, this is not as simple. A precision medicine cohort is not designed to capture the blood microbiome. It does not have the right controls, and there are potential artifacts, including sampling, wet lab, as well as computational, and we want to get the biological signal out of this. And this is quite a challenge. I won't go into the pipelines we had to design. We were, suffice to say, we had an advantage because we have data from different cohorts, as there were sub-cohorts in this study, and that allowed us to compare and contrast these cohorts to identify artifactual signals. And so this is work by Cedric Tan, who's now doing a PhD at the University College London. And we use this data to then ask several questions, including what microbial signatures can be detected in blood, whether there's indeed a blood microbiome, and where do blood microbes that we detect originate from. I must say this is also work along with Chia Meng Hao, who's a postdoctoral fellow in the lab. So The first striking thing that we observed as part of the study is that there's evidence for sporadic translocation in the gut microbiome, but not persistent microbiome signatures. So there's low prevalence of bacteria in blood. Most individuals have signal at most for a single species. Cutibacterium acnes, a common skin bacteria, is the most common species, but the rest of the species are quite rare. Less than 2% of individuals have many of these species. And then when we look at the potential origin of many of these species, where they've been reported before, many of these species seem to be common constituents of the human gut or oral or skin microbiome. So for example, here you can see several lactobacillus species, several bifidobacterium species, Faecalibacterium prosnitzi, and so on, which are well-known gut commensals. So we speculate that perhaps what we're observing are just signatures of sporadic translocation from other human microbiomes. In the case of the oral microbiome, this is a well-described phenomena, but also in the case of the gut microbiome, the leaky gut hypothesis has sometimes been used to suggest that this might lead to translocation of bacteria or bacterial DNA into the bloodstream. In addition, we want to look at and see if there is evidence for activity. And since we only had DNA sequencing data, we used an idea that was proposed a few years ago by Eran Elnav and Eran Segal, that we can use the coverage of the genome in a metagenomic data set to infer the rate of replication, because there's a skew in fast replicating bacteria. And surprisingly, we actually can see such signals. So in the case of several of the bacteria that we think are true signals, we can actually see a nice skew, while the contaminants tend to have relatively flat signals, suggesting that they are not from replicating bacteria. So while we see evidence for replicating bacteria, we perhaps do not see evidence that there is actually ecological interactions or a community of interactions, because when we look at correlations using this approach by Freeman and Alm, we find that while oral communities and gut communities have a large number of interactions that are predicted based on correlations, the blood microbiome is very sparse. And I say this fully aware that these interactions are not ecological, but they at least show that there's co-occurrence patterns which are similar to what we would see in a typical microbiome. And in the blood case, we do not see any co-occurrence patterns. This is partially predicted by the low prevalence of microbes that are seen in the gut, in the blood, so-called microbiome. So in summary, I want to highlight that whole genome sequences can be obtained directly from metagenomes with genome resolved assembly using long reads. And this is a powerful capability, especially for doing microbial surveillance studies, but also for harnessing biodiversity, microbial biodiversity, and uncovering novel enzymes. That multi-drug resistant bacteria seem to be enriched in hospital environments, and they seem to persist over extended periods of time to infect patients. That gut inflammation might be a key mechanism linking microbiome changes with CPE colonization, with multi-drug resistant bacterial colonization. And so both these reservoirs that we seem to be enriching need to be addressed if you are to address the AMR problem. And this is work led by many members of the lab, including Denny, Kern Ray, Jonathan Kang, Cedric Tan, Tarni Ghosh, and Lee Chen Hao. We are very grateful to have amazing clinical collaborators at the National Center for Infectious Disease in Singapore, including Kalis, Untek, and Barnami Young. From Tan Tock Seng, we have Brenda and Tim, and from NUS, Kanyon Wen, along with Yi Han Tan and Chiang Kai Cheng. And finally, we've been fortunate to be part of the MetaSub Consortium led by Chris Mason, and they've been great in sharing their protocols and collaborating with us. So I want to leave you with a final vision. We're hoping that we can actually go further and find ways to decolonize pathogens from various environmental reservoirs. we think the path forward is really to understand from a systems perspective how microbial communities come together. We are excited about the potential to obtain strains using high throughput single cell isolation techniques, use metagenomics to assemble genomes and understand their functions, and then to use co-culture experiments along with modeling approaches based on Lotka-Volterra models to really understand how microbes interact and assemble together, and thus identify inhibitors of pathogens that we can test in vitro models as well as in vivo models. So this is work that we are just starting based on a grant from the National Research Foundation here in Singapore. So we have open positions for postdoctoral fellows, students, and interns. So if you're interested in working here in sunny Singapore, we'd welcome all applications. So with that, thank you for your attention, and be happy to answer questions.
Thank you, Niranjan, for this exciting talk. So just a really broad overview of the things that we've been doing in the microbiome research area. So I really also like to hear about the environmental abundance estimates. So that was an interesting model. Let me see, we already have a question. So Sama wants to know whether there's, regarding the greedy contact assembly algorithms, hold on. Can you see those questions as well? Then maybe...
Yes. Yeah, I can see them.
Okay, whether there are any theoretical results that show that the resulting contexts are reliable?
Yeah, so that's a great question. The greedy contig assembly algorithms, as far as I know, do not come with any correctness guarantees. Often the assembly problem formally is formulated as some sort of traveling salesman problem or a tour problem. And if you want to do the contig assembly problem exactly, they're only under certain conditions where you have sufficiently long reads to span repeats and sufficient coverage of the repeats in your genome. And these are two constraints that we had established a while ago in a paper. Under those conditions, the parametric complexity of assembly is actually almost linear time. But those conditions have to be met. Without that, I do not know of any polynomial time algorithm that has been established, or any algorithm which has guarantees about the correctness of the assembly. There has been a field of work which looks into establishing whether a certain assembled region is of high quality or low quality. So there are regional quality estimates. Of course, there's base level quality estimates that give you a sense of how accurate each base is, but more importantly, the structural quality information is sometimes more important, whether there's potentially a rearrangement or a misassembly at a certain point. And there are methods which estimate that, but it's still not as actively studied as we would hope to.
Yeah, thank you very much. We have already some more questions. If you...
All right, so I guess the next question is, in the gut microbiome interaction, a model is fitted to a set of samples, that's right, which are clustered together. So the clustering is done before the fitting step. How's the clustering of samples done? Right, so sorry, I was not very clear on that aspect. The clustering, there is no clustering a priori. It starts with the full database, and in the first iteration tries to learn a model. Then it goes back and looks at the data points that are there, and then asks the question whether those data points seem to be coming from the model that it's learned. Of course, this is not done at every step, but at certain fixed intervals. And once it finds data points that don't seem to be coming from the model that has been learned, those data sets are thrown away, and the rest are used to fit the model. Of course, there's the risk that you might reach unstable equilibrium, or you might not, your data set may not have one model dominant enough to learn a stable equilibrium. So you might learn something which is intermediate between the two sets of points that you have in your database. But I think in practical data sets, there's usually a dominant enterotype, let's say. And so we find that our model learns that first, our approach learns that model first, and then we can take the points, the data sets that have been left behind, and learn a model for those data sets. So I hope that clarifies. So the next question is about how often do you see the AMR genes determinants in plasmids and bacteriophages? How do you link the bacterial genome with its plasmid from the assembled context? A very good question. So AMR genes, of course, can be everywhere. They can be in the chromosomal aspect and the plasmid, as well as in phages. In CREs, the carbapenem-resistant Enterobacteriaceae and Enterobacterialis, they're frequently in plasmids. In fact, that's the area of most concern, because these plasmids can be transferred from, say, Klebsiella to E. coli, and then you're just sort of spreading the plasmid and the resistance just through plasmids. And now, in terms of linking the bacterial genome with the plasmid, in metagenomics, shotgun metagenomics, that's of course challenging unless you have high-C data. So this is proximity ligation-based data. And then you can use reads that map to the plasmid as well as the chromosome, and an enrichment of such reads to help associate the plasmid with the nucleate genome, so the chromosomal genome. So I hope that helps. So the next question, sorry. Okay, so the question is about a question regarding the strain resolved assembly of OperaMS. Did I understand correctly that the strain resolution is mainly based on the clustering and the extension of the previously assembled short read context using long read information? Yes, so indeed, the strain resolution requires that the contexts have been differentially assembled by the short read assembler that provides the initial set of contexts that are put together by the scaffolding step. But what is unique is that we, in addition to the first step where we do clustering, and I didn't go into that detail, there's a second step where within a certain species where we have obtained a cluster for, we try to delineate the strains in that species. And for this, we also use the reference database that we have access to. And so this allows us to really look at the coverage information. So within the same species, look at contexts which might have differential context coverage, and thus get two finer grained clusters which provide the strain assembly. So then the scaffolding is actually done in those finer grained clusters that we generate. Yeah, so the question of what happens if the assembly, the short read assembly already produces consensus context, then then we are toast, right? So we rely on the assumption that the short read data is more accurate than the long read data. And so if you're not able to assemble the short read data into distinct strains, the error rate of the short read data is greater than the divergence of the strains, then that is essentially impossible problem. But of course, with newer technologies such as hi-fi data and direct assembly of hi-fi data, perhaps that can be addressed. And so I think it really depends on the kinds of data sets that you're generating. I hope I've covered all questions. I do not see any additional questions.
Yeah, I think so too. Thank you very much for your clear answers. I agree. So I have one more question maybe about the last study with the blood, with the microbes in the blood. So if you have these low biomass environments, it's always the challenge to distinguish truly present from contamination. And so can you tell us like how you did that? So how you ensured that this was a biological signal from blood?
Right. No, it's a very good point. And sorry, I didn't get into the details of the pipeline. So as I mentioned briefly, we had the advantage that the samples were actually processed in more than 5 different cohorts. And each cohort has its own sample handling, DNA extraction, library preparation, and sequencing modalities. And so what we could do is compare the patterns of the various species that we detected across the cohorts. And we used some simple heuristics which relied on the fact that a species which is only present in one cohort at sufficiently high abundance, but is at much lower abundances in other cohorts, is likely to be an artifact of the processing or the reagents or the sequencing of that cohort. So we compared and contrasted signals across the different cohorts to hopefully sensitively identify um, signals which were really from contaminants from one cohort, right? So that was the approach. And then the second step was that in addition to using this approach to flag certain species, we also looked at the abundance of these species and how they correlated with the um, the the DNA that was extracted, or the library quantification that we did. And often contaminants have a negative correlation. So the lower the amount of DNA extracted, the higher the abundance of the contaminant in that library. So anything that had that signature was also provided as some information that potentially that's a contaminant. And the last signal is really that even if one of the contaminants was not detected by either of these methods, contaminants usually have high correlation with other contaminants. So we can take one species which has been identified as a contaminant and look for correlations with other species. And when two species are highly correlated with where one of them is a contaminant, we can flag the other species as a potential contaminant as well. So we use these approaches to try to be as sensitive as possible. I'm not claiming we are perfect, but I think there's a lot more results in the paper, which I hopefully convince you that majority of the signals we observe are not likely to be contaminants.